Hello, my name is Ralston Smith, and this summer I have been working on a research project in Dr. Solomon's lab. The title of my project is Continued Development of a Novel Granzyme B PET Imaging Agent for the Assessment of Cancer Immunotherapy Efficacy. Granzyme B is my target enzyme, and it is an important regulator of T cell and NK cell induced apoptosis or programmed cell death. Granzyme B is a protein that catalyzes the cleavage of proteins after an aspartate residue, and has an active site catalytic triad comprised of histidine 57, aspartate 102, and serine 195. The general amino acid sequence IEXD has been determined as a highly specific Granzyme B binding peptide, where X is a variable amino acid. Therefore, I have been working with the following 5 peptides to characterize their binding to Granzyme B both computationally and in the lab. Computationally, I ran docking simulations for each of these peptides with Autodoc Vina using the Hopper cluster at GMU. In the lab, I ran SPR and inhibitor screening assays to characterize binding. Surface plasmon resonance, or SPR, is an optical method of detecting binding of a small molecule to a receptor. This method can be useful for characterizing protein-ligand interactions. In an SPR experiment, all interactions occur on the surface of a sensor chip. Binding events cause changes in the mass on the surface of the sensor chip, which causes light to reflect off the surface at a different angle. This change in the angle of reflection can be detected and measured. One major drawback of SPR is finding the optimal parameters under which to run each experiment. This is computational data of the binding of each peptide variant to Granzyme B. I also included peptide variants with a metal chelator known as NODA that will serve as the imaging agent. Importantly, the energies of binding appear to decrease with the addition of NODA. This indicates a higher Granzyme B affinity of the NODA peptides compared to the untagged peptides, which is good. This is data from one of my SPR experiments testing IEAD. I used a program called TraceDrawer to analyze the data. The curves are not ideal, and they only seem to show an on rate for some of the curves here, with no associated off rate. Furthermore, these signals are very noisy, making it difficult to discern much information from the data. One way to make the data more readable is to optimize the experimental parameters. This is SPR data that I collected for IEGD. Clearly, the noise is still relatively high in this example. However, there seems to be an on rate and an associated off rate for some of the curves. Here are the peptide equilibrium dissociation constants from the previous SPR experiments. According to the data, IEAD has a much higher affinity for Granzyme B than IEGD because it has a much lower dissociation constant. However, the uncertainty in the data for IEAD is not within an acceptable range. Therefore, the experimental methods need to be refined. To the right is an example of the inhibitor assay data collected for IEFD. In these experiments, data has been collected for both IEGD and IEFD. The data so far has been largely inconclusive, however. I suspect that the lag time between loading the samples into each well of a 96 well plate and then loading the plate into the plate reader is a major cause in the ambiguity of the data. This lag time may have caused the fluorometric readings to have been taken after the completion of the reaction. One way to reduce this lag time would be to use a multi-channel micropipette for loading the samples. My next steps include troubleshooting my experimental protocols and collecting further SPR and inhibitor screening assay data for each peptide. This data will indicate the best Granzyme B binding peptide, and then I can move on to synthesizing this peptide with a NODA tag and characterizing the binding of that molecule to Granzyme B. This is NODA. I would like to thank Dr. Lee Solomon for his ongoing support and leadership on this project. Thank you to Dr. Foreman for sharing his expertise in computational data collection, Dr. Veneziano for providing access to his SPR instrument, and Dr. Michael Page for providing access to his fluorometric plate reader. Thank you Dr. Karen Lee and the Oscar URSP for funding this project. A special thank you to Dr. Oluwatayo Ikotan Assistant Professor in the Department of Molecular and Medical Pharmacology at UCLA, for inviting us to work on this project. From Dr. Solomon's lab, thank you to Robert Spain, Jacqueline Yasa, and Annie McAllister for their helpful insight into both computational and lab work. From Dr. Page's lab, I would like to say thank you to Zach Bulak for providing specific laboratory training. Thank you to Matthew Jacoby for providing example scripts with explanations for the Hopper cluster. And finally, thank you to Elena Benson for assisting me on this project this summer. Here are my references. Thank you for watching.